Hello, and welcome back to Introduction to Genetics and Evolution. So far, we've studied the inheritance of very simple genetic traits, traits whose variation is caused by maybe two alleles at a single locus, and we've looked at how to map that genetic variation. What I'd like to do now is get into some slightly more complicated facets. Let's look at how traits really are. Most traits are not, or most variation in most traits is not usually caused by variation at a single gene with two alleles. So we'll first talk about mutations as the source of all genetic variation, and then we'll go into what the genetic basis is of normal traits, and what are some complications rather than the overly simple model we used so far. So let's get started. If you go back somewhere between 3.5 and 4 billion years ago, we had what we believe to be the first origin of life. Now, we don't know exactly what that life looked like or exactly the conditions were, but we can imagine that maybe there was something along the lines of a single haploid genome. When I say haploid, I mean having one copy of the genome, and a single primitive cell. Now, genome there was probably an exaggeration. It probably wasn't anything nearly as complicated as most of the genomes you see today, but it would have been some small piece. Now, the question I like to pose to you is, what would have to be true for you to define this thing as alive, this original cell as alive? How would you decide it was alive rather than inanimate? Well, the most likely answer people would say is it replicates, it reproduces, that it makes another copy of itself. So this one makes these two other daughter cells. And what it makes will be dictated by its gene. Now let's imagine hypothetically that the genetic code that was found in that original cell never ever changed over the 3.5 billion years of time. What would life look like on the planet today? Well, the answer is if it never changed and if its genetic code is dictating what it can make, basically life would look like this today. There'd be a lot of copies of that simple haploid genome in cells all around the planet. That's obviously not what we see today. So there obviously has been introduction of some sort of new variation into the diversity we see on the planet today. Now, where does this variation come from? Well, this is the theme for today's lecture. Where does this variation come from? Well, what is the source of these changes in the genetic code? Well, the answer is mutations. Mutations are often defined in textbooks as a change to the DNA sequence, but very importantly, I'd like to emphasize they are the ultimate source of all genetic variation on the planet. Because again, if we have a single origin of life, which we do assume, a single origin of life, common ancestry of all existing life, then all the diversity we see has to come from iterations of mutation from that original self-replicating form. Okay? So what is a mutation, or what is a mutant? I mean, you hear this word very often, both in common parlance and in the movies, things like that. Well, a mutant may be what you call your brother when he's being very annoying. Get off me, you mutant! I've probably heard that many times. Those of you who are fans of science fiction, a mutant in the comic world is an individual who possesses a genetic trait called an X gene that allows them to naturally develop superhuman powers and abilities. So there's a picture here from the comic X-Men. Many of you may have seen the movies from that. Probably not the definition we'll be using here in this class, as you'd guess. You may think of a mutant as someone who has an extreme disfiguration, such as extra fingers. So this individual right here has polydactyly. So if you count, they actually have 6 fingers. This is actually something that does come up. This is a mutation that does crop up periodically. It's a little harsh to refer to the person as a mutant because they have this, but it is a mutation which arises. Now, honestly, there's a little bit of truth to all those definitions, though less so for obviously the X-Men version. But again, the ultimate source of all variation is mutation. It is any sort of change in the genetic code. Okay? So you can think of it this way, that mutations happen. And they happen a lot. They happen far more frequently than you may guess. And you'll see in a subsequent video we'll talk about mutation rates. Now, one of the most common types that we'll focus on for this is an error in replication or in meiosis that leads to a change in a base, or a nucleotide. So imagine you have this stretch of sequence in a mom, okay? And let's say the mom is homozygous for this. So she has 2 copies of this stretch of sequence that starts with ATG. Now let's say in one of her eggs that makes the kid, there was this base change, went from A to G, such that this is actually what's in the kid. The kid then has a mutation. He or she has a nucleotide variant that was not found in either of the parents. So that is by definition a mutation. Now, importantly, let me emphasize what a mutation is not. So mutations do not happen when you need them. Right? They don't happen preferentially when you need them. You can imagine something like, oh, it would really be great to have 6 fingers. No, that doesn't happen at all. They are random with respect to need. Okay? Now, many mutations are bad. In fact, probably you could argue that most mutations even are bad, or at the very least, a large fraction. Now, why is that the case? If you think about it, our genetic code is not completely, but largely fairly streamlined. So if you introduce a random change, it's likely to be something that's bad. It could be something that doesn't really matter, but it's likely to be something that's bad. The analogy I often like to use is imagine that you have a car, right? And this car has come together over a very long period of time. And you take a hammer from way across the room and throw it towards the car. What are the odds that that hammer will make the car better in some way? It's probably not 0, but it's extremely low. In contrast, what are the odds the hammer will either do nothing, or nothing substantive, or it will actually be bad for the car? Probably pretty high. So on average, we tend to assume that most mutations are either bad or they don't matter. Okay? I use one example here of one that doesn't matter. So many of you are familiar with the triplet code. So again, genes tend to code for proteins. They have a triplet code associated with it. And very often that third base in the code does not change which protein is produced. So if you have, as part of this code, AAA versus AAG, you would actually put the same amino acid into that protein. There actually would be no change in the final protein. So again, most mutations tend to fall into one of these two categories, either the bad or the don't matter. Now rarely, but it does happen, a mutation may be good, advantageous. Now, well, again, it doesn't arise when you need it, but it may just happen to be good. And it may not necessarily even be good at the time when it first arises, but it might end up being something that is useful in a subsequent generation. So thinking about that, let's imagine that a mutation has happened, and the new variant now exists in the population. So here's our first generation. Everybody has the same sequence. Okay. Now we have this one mutation, like the one I talked about previously, which is arisen in one egg cell. Now, as you'll notice, we now have the birth of a SNP, a single nucleotide polymorphism, because here all the sequences were exactly the same. Now we have one site which is polymorphic. It has 2 different variants there. Now let me ask you a question. Is it more important that this is transmitted meiotically or mitotically? Remember we talked about meiosis and mitosis way back in the beginning. Well, the answer is definitely meiotic, because if let's say for example this new mutation starts in this part of your hand, it spreads to the rest of your hand, it's never going to get passed on to the offspring. It's much more important it gets transmitted meiotically so that it gets on to further generations and gets into generation 2, etc. So what happens then? What happens then is this new variant may be lethal. It may cause immediate death of the embryo. Right? That's a possibility. In the previous slide, I showed you one individual was heterozygous for it. Maybe that individual will die at the age of a couple of months in utero. It could cause some other sort of loss of fitness. It could be, yes, it doesn't immediately die, but they have extreme, gastrointestinal distress that makes it so they're not able to thrive as well. That's a possibility. These are definitely in the bad category, right? Now, it could be that this mutation causes no change in fitness, that this mutation is just the same as the parent in terms of overall probability of passing on the genes. So this would be what's referred to as the neutral category. And finally, maybe it causes superfertility. Maybe it makes it that this person is extremely attractive, or that when they do mate with an individual, they're much more likely to fertilize. This would definitely be the good category. And this would be much more like the X-Men version, presumably. So let's follow what happens after that. So again, we left off last time with generation 2. Here's this new mutation. Well, it may spread, it may get lost. It's possible that this individual would leave no offspring. Let's imagine this the case where I said this individual died at a very young age. Or it could just die for a random reason. Maybe this individual gets hit by a car at the age of 10. It could happen that this just never gets transmitted. That's one possibility. But let's assume it does get transmitted. Well, then, some individuals get it. This individual gets it here. This individual gets it here. In this case, we even start getting some homozygotes because this individual gets it and passes it on and has a kid with this one. So again, we have an individual here who's homozygous. We have a few others. This one seems to be spreading in this particular case. So it looks like maybe this particular mutation was advantageous or possibly neutral, but certainly not very bad. So this is the kind of thing that can happen with new mutations. So we have this new variation, it arises, it may just get immediately lost because it's bad. It may get immediately lost if it's neutral. It may even get immediately lost if it's good. Let's say, for example, this individual had super fertility, but again got hit by a car. That could happen. But for a lot of mutations, they will arise. You have the birth of a new SNP. They will be there in a population for some time. Now, over time, many rounds of this mutation, not just that individual one, will generate a lot of variation in natural population. And again, as I emphasized, a lot of mutations don't necessarily have an effect on fertility or lifespan, but they may still affect a phenotype. They may still affect how an organism looks, right? So let's say, for example, there was a mutation that slightly lengthened your earlobe. Maybe that wouldn't affect your probability of having kids whatsoever passing on your genes, but it still could get passed on. And in that sense, it is still neutral, even though it has an effect. Now, I'm going to come back to this example in the next video, but I just want to introduce it. There's lots of genetic variation in things like, for example, human height. Now, some of that, of course, has to do with nutrition rather than genetics. But where does that variation come from? Is that actually all just from mutations at one gene? Well, take a look at the next video and see what you think. Thank you for joining. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about mutations as the ultimate source of all genetic variation. But at the very end, I posed a question to you. When you look at a lot of traits, we see a lot more variation than seems to be explainable by simple single gene, two allele inheritance, right? That if you look at something like human height, even after you correct for things like diet and things like that, you tend to see a lot of variation. There are some individuals that are very tall, some individuals that are very short, and a lot of people with sort of quote-unquote intermediate heights. And that's not true just for human height, but it's true for a lot of traits. So why do we see so much phenotypic variation? And it's phenotypic variation that we know often has an underlying genetic component. Well, the answer to that is that the simple single gene, two allele model is actually not sufficient for explaining the variation that we see out there. And in this lecture, we'll go over several reasons why that's the case. We'll focus on 5 particular things. But the big question that I want you to think about to start with is why do we see so much variation in traits? At the very end of the last video, I posed to you the question of height as one example, right? Well, one possible explanation, this is not necessarily a complete explanation, but one possible explanation is that more than one gene controls variation at a trait. So something like human height, rather than having a single gene that has a tall allele and a short allele, and you could be homozygous tall or homozygous short or heterozygous, maybe there's a lot of genes that contribute to variation in height. Now, honestly, this idea of more than one gene controlling any trait is probably true for just about any phenotype you could study. Like, there is no single diabetes gene, there's no single cancer gene, things like that. You may hear these kinds of words all the time in the media, but they're just not true. Now let me give you a fictional example. Let's pretend that alleles at 6 genes control human height. Okay? Let's look at what this would result in. Now here's a simplified example. Let's imagine there's 6 genes for women's height. We'll focus specifically on women's height. Okay? Now let's say that for each case, look at the bottom of the slide here. For each case, your height in inches is determined by your genetic code, and you are 5 foot 0 inches plus the number of capital letter alleles in inches. Okay, so imagine that. I apologize for those of you who are used to the metric system. So you could do the same thing with meters in some way. But let's say that based on the number of capital letter alleles you have, you add 1 inch for each one. So let's try that with the first one here. Let's just count the number of capital letter alleles. Well, for the first gene we have 2. Second gene we have 1. Third we have 2. Fourth we have 1. Fifth we have 1. So we would say you're 5 foot 0, 5 foot 1, 2, 3, 4, 5, 6, 7. So this individual would be 5 feet 7 inches tall. The next one has 2 capital letter alleles, the B and the D. So this individual might be 5 foot 2 inches. Okay. So let's imagine that you iterated this across all these different things. And this is, again, a completely fictitious example. And we're assuming now each of these 6 genes only has 2 alleles, has a tall variant and a short variant, and they all act somewhat independently of each other. Okay? So what would you see? Well, we can iterate across all these different genes and all these different people. And this is the spread we see: 5'7", 5'2", 5'6", 5'8", 5'6", 5'6", 5'10". Now I want you to look at this distribution for just a minute. We have 3 different individuals that are 5'6", but if you look at them, none of them actually have the same genotype. Right? If you look at them like this individual right here, individual number 6 has a capital A, and a little a. Individual 5 has 2 little a's. Compare individual number 3 here has 2 capital B's. Individual 5 has 1 capital B, and individual 6 has no capital B's. So there's a lot of variation, but it ultimately leads to the same height. So as you can see from this model, there's a lot of different ways to be 5'6". In contrast, how could you be 5'0"? There's only one way you could be 5'0", and that would be if you're lowercase for all of them. So what this does, if you iterate this, is you come up with this sort of a distribution. Right? You come up with what is referred to as a normal distribution. It's not intended to imply that other distributions are abnormal, but just this is something that you see quite a bit, that there's a lot of different ways to be intermediate, but there are fewer ways to be extremely tall or extremely short in this particular model. So this is often called also a bell curve. And again, there's only one way you can be 5'0" or 6'0", but there are many ways to be 5'6", and that's why you see this distribution like that. Now, one example, if this is one that some of you may be more familiar with, is it's just like test scores. There's a lot of different ways you can get a 50% on a test based on which questions you get right or wrong. There's only one way to get 100%. There's only one way to get a 0%. And those are get everything right and everything wrong. Okay? So what happens from this is you can generate what looks like continuous variation from variation at many genes. And this is even though each of the individual genes are inherited in a Mendelian way. So let me break this down with a couple of sub-examples. So you could do the same sort of thing if you do an F2 cross. An F2 cross is the same as crossing 2 inbred types. So for example, big A's to little a's, taking the F1s, which we recall are the offspring from that, and having the F1s breed with each other. That's the F2. So in this case, if we have one gene contributing to variation in height, and you start with these homozygous tall, big A, 6-foot kids, breed them with homozygous short, little a, 5-foot tall individuals. So all the offspring would be big A, little a. Right? So let's say we cross big A, little a to big A, little a. Well, we do our little Punnett square that we're very familiar with. Right? And in the F2, we get 1/4 big A. We get 1/2 big A, little a, and 1/4 little a. Right? There's 2 out of 4 ways to be big A, little a, 1 out of 4 little a, 1 out of 4 to be big A. So you get something kind of like what we just saw. This is now from just one gene. What if we did 2 genes and they sorted independently? Well, if we did 2 genes, and then let's say, for example, it's this time it's big A, big B versus little a, little b, then you'd have a slightly broader set. You can follow what's going on in the A gene, follow what's going on in the B gene, and you'd get 1/16 would be little a, little b. 1/16 would be big A, big B. And then you'd have a whole bunch of other intermediates that then would have the different sorts of heights. This would be the one that would be 5 foot tall. This would be the individual that would be 6 foot tall. But all the rest would be intermediate. And this is, again, the spread that you see. And finally, this is the example we actually saw, that if you have 6 genes for height, you generate this virtually normal-looking distribution, or bell curve. It's continuous and it's bell-shaped. It's not exactly continuous, but it looks pretty close to continuous in this case. So that's pretty cool. And this shows you how variation at more than one gene contributing to a trait can generate this sort of variation that we see in traits out there, where things aren't just tall versus short, but they're much more continuous. So why else do we see so much variation? One other possibility is what's referred to as variable penetrance. And this is the case of what happens when mutants don't always affect the phenotype in the predicted way. So let me show you an example of this. The eyeless mutation in Drosophila is one that exhibits variable penetrance. Now this, on the left-hand side here, we have a Drosophila with a regular eye. There it is. So it's a nice, big, beautiful thing. When you have the eyeless mutation, you often have a shrunken eye. So you see this individual has a very small eye. Now, what's tricky is you may be homozygous for the eyeless mutation. You may have 2 copies of it. Sometimes you actually will have a normal eye. Sometimes you'll have a shrunken eye like this. Sometimes you'll have no eye at all. It's always the same mutation, but it doesn't always have the same effect. And this is why we say that mutation is not fully penetrant. It exhibits variable penetrance. Now, this is very often true when you look at disease susceptibility genes. So, for example, the BRCA1 breast cancer susceptibility gene that's out there, in most of the population, if you're a big B or a big B, little b, you have a low chance of getting breast cancer, on the order of 12%. Unfortunately, that's low. If you have 2 mutant alleles of this gene, though, then you have a 60% chance of getting breast cancer. So this is 5 times higher chance of getting breast cancer. This is 5 times what is a reasonable risk there. But not everybody who's little b will get breast cancer. Right? This is not fully penetrant in that regard. So the BRCA1 breast cancer phenotype is not fully penetrant because you can be little b and still have all non-cancerous cells in your breast. So this sort of variable penetrance also contributes to some of the variation we see in traits. Okay? Well, what else could do it? Well, another very interesting one is the idea of interactions among genes. Right? So essentially what's happening in terms of your genotype at one gene will affect how much the other gene contributes to the trait. So let's go back to the example I showed you earlier with height. So in this example, we used basically all these different things as completely independent of each other, and we just added up their effects. So we refer to this as sort of an additive model, where we just add up the effects of what's going on at all these different genes at A, B, C, D, E, and F. Well, in fact, that's not always the way things work. Often, especially when you have things like pathways, you can have epistasis. So here is the example of pea flower color. Now, most pea plants will have a nice pink color to them. And there are several steps in the production of this pink color in them. And these steps are genetically determined. So let's imagine you have this precursor. This is the compound that ultimately will lead to the pigment in these pea plants. Now, there are 2 genes. The C gene and the P gene produce the C protein and the P protein. And these lead through these intermediate steps to ultimately make anthocyanin. Anthocyanin is what gives them this beautiful pink color. Now, you can actually stop this process at either place. If you are homozygous for mutant at C or homozygous for mutant at P, you don't produce any anthocyanin, and instead you end up with a white pea plant. Okay? So let's follow this through here. Now, we can block the process at either place and get white peas instead of pink peas. So what this means is if you're homozygous little c, or little p, it doesn't have to be both, then you will produce white peas. Otherwise, if you have at least one capital letter allele, the dominant normal allele, then you'll produce the beautiful pink peas you're supposed to produce. So here's all the possible things you can do. Let's go through each of them and label whether the peas would be pink or white. So this one would be pink, 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 pink. This one has 2 little peas. So that stops the process right here. It would get from precursor to step 1, but you never get the anthocyanin. So this would be white. This would be pink. This would be white. This would be pink, pink, white, white. These ones over here stop at the C. Pink, white, white, white. 1, 2, 3, 4, 5, 6, 7. So there's 7 different possibilities that lead to white one. Now this actually was observed by William Bateson and some of his colleagues many, many years ago, where they actually found in doing this particular cross, they found a 9:7 phenotypic ratio. Now, this didn't seem to follow standard Mendelian inheritance, and he didn't immediately know what was going on. But they inferred that there was something like this going on, that essentially there were variation at one gene was affecting what happens with variation at the other gene. So look at this example right here. Now normally, again, capital C and capital P are supposed to work together to lead to a pink plant. So if you look at this example right here, if you are little c, it doesn't matter what your genotype is at P. You will always produce a white pea plant. Similarly, if you're a little p, it does not matter that your genotype is big C over there, because you will only produce a white pea plant. So in that sense, what's happening at one allele, or I'm sorry, at one gene, is moderating whether or not variation at the other gene even matters. Okay? So that is epistasis, because that is an interaction between the two things. It makes it so what's happening at one gene doesn't matter. We didn't see that with the height example because at that point we had to account for what was happening at every single gene. Here, once you see the little p, you don't even have to look at C. Okay, so that is epistasis. Let me give you another example. This one, instead of from peas, is from mice. This is what produces mice with this nice sort of brown, what's referred to as agouti color. Again, there's two genes that contribute to it, and you have to have both the A and B to get it. In this case, there's a slight deviation from what I talked about before. If you're little b, you actually produce albino mice, these all white mice. Okay? In contrast, if you're little a, but big B, then you actually make black mice, like this one. You don't make the agouti ones or the white ones. So what happens here is what's going on at B moderates the effect of A. So A changes you from agouti to black if it's with a big B. In contrast, if you're with little b, then what's going on at A doesn't matter because you're always going to be albino. So again, you have this masking of the effect of one gene. You're masking what's going on at A because of the genotype at B. So that is an example of epistasis. And honestly, that's only one way that epistasis can happen. Two genes where gene 1B is a switch that turns off gene 2. Right? This is similar to what we just saw. Whereas gene 2 affects the deposition of coat color, where one causes black and the other causes brown. But if you turn off the switch, it doesn't matter. Right? So this is what was going on in this particular example. The genotype at B modifies the effect of the genotype at A and sometimes eliminates its effect completely. Okay? So epistasis is something that, again, can generate unexpected diversity and is one of the complications to simple single gene inheritance the way we were following before. Well, what else can happen? Well, there can be more than two alleles at a locus. Almost everything I've shown you so far has had this very simple big A, big B, little b. But in fact, there are times when you have more than two alleles at a locus. Probably the most famous example of this is this classic example of A, B, and O blood types. Now, these are related to variation at a gene located on chromosome 9 in humans. There are three alleles, A, B, and O, as you see from the title. A and B are both dominant over O, but A and B are not dominant over each other. And what happens in this case is A and B create very specific antigens, but O does not. So the possible blood types you can have are A, B, AB, and O. And the possible genotypes are these: AA, AO, BB, BO, AB, OO. Now I said A and B are dominant over O. So what if you are AO genotype? What blood type would you be? The answer then is you'd be A. So AA and AO both have blood type A. BB and BO both have blood type B. A and B are not dominant over each other, so you just be blood type AB. And OO would obviously just be O. Now, from the medical angle, if you get a transfusion with the wrong blood type, you will actually reject it if it has a foreign antigen. So if you're an O blood type, it's very good for you to donate blood because you don't have any of these antigens. You don't have the A or B antigens. Anybody can get O blood transfusion and they'll be okay. In contrast, if you're AB, you're the best for receiving a blood transfusion because you have both antigens. Doesn't matter what they give you, you'll be okay. So that just illustrates the medical relevance to this particular example. Now, what do you do when you have these three genes at a locus? Well, again, as I said, A and B are dominant over O. Here's all the blood types. Basically, the inheritance works just the same as the way we studied for single genes. So I did a cross here as an example of an AO female crossed to a BO male. And you see you get four possible blood outcomes there. You can be AB, BO, AO, or OO. Let's do one just as an example. Let's say that you crossed AO to AO, right? So let's do that. AO, AO. What would you get? Well, you may get AA, that would be blood type A. AO, which would be blood type A. AO, which would be blood type A, or OO. So if you have the genotype AO, your wife has genotype AO, you have kids, on average, a quarter of them should have blood type O, 3/4 should have blood type A, just like you and the mom. Okay? We just followed basic Mendelian inheritance. The only difference in this case is we actually have 3 alleles rather than 2. So we've gone over 4 different ways we see so much variation. That you can have more than one gene controlling trait. You can have variable penetrance of one or more of the alleles. You can have interactions among genes or epistasis. There can be more than 2 alleles at a locus. But the big one that we didn't really talk about, and this is something we'll come back to in some of the later lectures, but I just want to mention it briefly here, is obviously the environment and interactions with the environment. Now, when we come to heritability, we'll talk about this quite a bit. But this is one of those things that is probably really obvious. The environment can affect phenotypes. It can either affect them directly or they can interact with genotype. Probably one of the simplest examples many of you have experienced is sun tanning. In the absence of sun, you don't tan. So clearly that is an environmental effect. And I'm not talking about tanning beds, of course. And also, many of you probably know that some people naturally tan more easily than others. Right? That there are some genotypes out there that will tan very easily, and some genotypes that won't tan so easily. They're much more likely just to burn or to freckle or something like that. And that is an interaction of the environment with genetics. So that gave us, again, 5 different ways that we actually see so much variation out there. And this is why simple single gene models are not typically sufficient when we're trying to understand genetics. So in the next video, we'll talk briefly about mutation rates, but then I want to come back to how do we map traits when they are complex like this, when they exhibit things like potentially epistasis, having multiple alleles, having many genes contributing to them. We'll come back to that shortly. Thank you very much.